So we have a very exciting lineup here, 2 theorists and an experimentalist, and we're going to get to see a pretty exciting slice through control and neuroscience. And so I don't want to take up much time. So first we have Rodolphe Sepulchre, who is from the legendary controls group at Cambridge. And he's going to talk about what I call levels in the in our neural control systems. So Rodolphe.
Thank you. So that was my first slide. A new home for my research, how to design reliable neuromorphic systems out of uncertain transistors. Control is design of uncertainty. And then the 2 historical moments of control, Maxwell with the new hardware being the steam engine, and then Bell Labs in the 30s and 40s, the new hardware being amplifiers. And I was saying that perhaps we are in a similar situation with trying to build these neuromorphic computing systems out of uncertain transistors. And I said that, okay, I came to Telluride last year, after studying neuromodulation and the control theory of neuronal circuits, in particular the work of Yves Marder, who has been studying neuromodulation in a specific network for more than 40 years. And as you might know, Yves Marder got the Kavli Prize in Neuroscience in 2016. And I think the citation of the prize is very telling, especially for a control theorist. So it says Yves Marder used simple circuits to elucidate the dynamic interplay between flexibility and stability. I was saying reliability and robustness. She showed that numerous neuromodulators reconfigure the output of circuits without altering their underlying anatomy. At the same time, she found that circuits can generate similar neuronal and network output from many different configurations of intrinsic neuronal excitability and synaptic strength. So I've been fascinated by these control principles in neuronal systems. And I think I came last year with a question that I wanted to probe as a participant, which is neuromodulation relevant for a control theory of neuromorphic systems, especially when we want to address the issue of uncertainty and of maintaining reliable outputs in the presence of uncertain components. So what I plan to do today, and I apologize for the bad start, but is a sort of a brief summary of what I presented last year, which was a very specific neuromorphic circuit and an attempt to implement neuromodulation in silico. And then I would like to broaden the scope and perhaps share for about 10 minutes what I think are the main questions of what I would call neuromorphic control. And perhaps finishing with a proposal or a start of a proposal for a challenge that could be proposed next year. And I would be very keen on getting feedback and suggestions. We still have some time to fine tune the details. So I came last year at the end of the PhD thesis of my student, Luca Ribar, and I think Luca is in the audience today. Luca spent 3 years designing a single neuron in silico that sort of imitates the principle of neuromodulation. And there is a specific single cell mechanism that we want to replicate in silico, and which is illustrated here by 2 experimental traces, single neuron recordings, and one in the thalamus and the other one in the subthalamic nucleus. Those types of neurons, show up especially in many areas of the brain, but in areas that involve gating sensory inputs or gating motor outputs. And what you can see here is that under inhibition of the cell, the excitability of the neuron, so the sort of single cell excitability changes dramatically. You go from fast tonic spiking to slow bursting to equilibrium or silence, but in fact burst excitability. And what a main outcome of Luca's thesis is to build a very simple neuromorphic circuit that reproduces exactly those same features. So if you apply a current, then you can start from a current level where you have fast tonic spiking, and then you, under inhibition, you go through this bursting pattern. And then if you further inhibit the neuron, it becomes silent, but a small excitation produces a burst. And the claim in Luca's thesis is that you can make this circuit very reliable, in the sense that even if you have vast uncertainty on the transistors, you still will be able to tune quite easily and to reproduce that same pattern. And then you can really tune, deform as you wish the feature of the bursting or the feature of the spiking, and so on. And the structure of this circuit is extremely simple. It's made of a parallel connection of current sources, and each current source just use two transconductance amplifiers in the linear regime. And you use one of the amplifiers to set up a time constant for the activation of the current, and another amplifier to set up the gain, the sigmoidal gain of the current source. So you have a current source that is voltage dependent, and that mimics the current, the intrinsic current produced by a particular type of ion channels. And the control of the circuit is consistent both with neuromodulation from a biophysical viewpoint, but also very consistent with the hardware, in the sense that you just need to control the base currents of those transistors to be able to tune the maximal conductance of the current. And that's very often how you model neuromodulation, because biophysically neuromodulation occurs by changing the density of ion channels and by modulating the density of receptors, and all of that can be modeled in a crude way by changing the maximal conductance of a specific current. So that is the control architecture, and then this is a picture of the circuit that shows you how ignorant we are from neuromorphic hardware, but it still worked quite okay, and Luca was able to reproduce experimentally predictions from the principled design that he was proposing.
So Rodolphe, just to interrupt you here for the first time, if you go back to that thing, when you, I'll just draw on your screen here. So what's changing here, right? What's changing here? Some conductance or some input current?
Oh, on this particular, so this is just an input current. And so as you increase the input currents, you modulate, you go from what is called slow spiking to bursting. So that would be something that is reminiscent from, modulating the applied currents, and you go from, it's a different pattern, but here you go from fast spiking to bursting. Okay, so you change the pattern just by applying an external current. Yeah. Okay, so I think that when I presented that last year, it was sort of a sanity test for us to check against an audience of experts, whether we were just 10 or 20 or 30 years late, because of course many developments of neurons in silico have been proposed before. And of course, beyond that, we wanted to probe whether this was possibly relevant for neuromorphic engineering, and whether this could be called probably a neuromorphic circuit, and whether there was sort of a consistency between the biophysical idea of neuromodulation and what you could implement with the existing hardware of neuromorphic chips. And I think that, the response was really great. Not, I think, just because of the specific atmosphere of Telluride, that people are so open-minded and so patient when you are a newcomer. So if you are a newcomer to Telluride, don't be afraid. You will. I got fantastic feedback, very positive criticism, and I learned a lot. once again, the emphasis in our work compared to the previous works on designing excitable neurons in silico was on the ability of having a principal tuning of the maximal conductance, which is consistent with what is known from the biophysics of neuromodulation. And to show that you could start from very sloppy components and that it doesn't really matter. So it doesn't rely on a very precise parameter tuning, because what you are tuning at the end of the day is the input-output conductance of the neuron, which is very much how a biophysicist thinks of the neuronal membrane. You are not tuning the parameters of a particular state space model or particular differential equation that models your neuron. And that's often the source of a lot of fragility. Okay.
But why are you not doing that? I'll just interrupt you again, because basically you're changing the conductance, which changes the time constant, which is essentially the same as changing a parameter of your differential equations. And you have the additional complication that the transconductance amplifiers saturate, and the saturation is exactly where all the instability usually comes from. Usually the limit cycle oscillations that you can't control come from that saturation effects in the feedback. Yeah.
So what I'm contrasting here is really input-output tuning versus tuning of a state space model. Does that make sense? This difference?
No, I don't know the terminology.
So even if you go, I mean, that's a very classical issue in control, in linear control. The input-output model is, for instance, a transfer function, versus the state space model, which is a differential equation, which is a specific realization of the input-output equation. And it is well known that when you want to address robustness, you really prefer to work in the input-output framework. But then when you have to translate that into actual computation, you do that in the state space framework. And a lot of the theory in control has been to develop a bridge between those two worlds. Okay, but robustness and uncertainty is really, it's much better to study robustness and uncertainty in the input-output framework. It's very difficult to match the uncertainty of a parameter in a state space model to what it will lead in terms of input-output properties. Okay, so that's already true in linear.
I presume that if you want to, if you want to understand that, look at your paper, you know, because that's where you discuss these stabilities, right?
Yes.
Yeah, it'd be good if you put the link specifically to that paper in the Slack.
It's an issue that is already present in classical linear theory, and it's even more so when you deal with nonlinear state space models, because it's very difficult to predict when you change a parameter. Of course, you can apply bifurcation analysis, but there is it's not an easy match and it's very hard to handle uncertainty at the level of the dynamical system itself. And that is the source of fragility that you see in lots of models of the literature. Okay, now just to give you a little update on where we stand 1 year later in this particular project. So I'm very grateful to Gert Koenbergs, who was a participant last year and who has designed neuromorphic chips for 20 years or probably more. And so what we have been doing this year is to sort of replicate Lucas circuit in an existing neuromorphic hardware, namely the Neurodin. And Neurodin, for those of you who don't know what it is a chip that essentially consists of 4 neurons, and each of the neurons implements Hodgkin-Huxley types of currents, a pair of currents, and then you have you can digitally design the synaptic interconnections. Now, what we are doing here in our circuit, we use pairs of currents that very much resemble the Hodgkin-Huxley currents. So we can design one pair of these currents with one neuron of the Neurodin. And then we can shunt 2 neurons to implement our second pair of currents that we need to generate what we call slow excitability, the bursting pattern. So essentially we shunt 2 neurons of the Neurodin chip to generate one bursting neuron. And then we use one synapse to create the inhibitory connection of the half center oscillator. And so the Neurodin chip is a perfect platform to implement what is possibly the simplest neuronal circuit, which is the half center oscillator, the inhibitory connection of two bursting neurons. Okay. And what we wanted to demonstrate on the Neurodin chip is that we could apply our design principle and exhibit neuromodulation. Now, this has been very successful, and unfortunately, it has been slightly slowed down by the COVID epidemic. So we haven't been able to complete the experimental results, but I think we are very close to have experimental results that will confirm that that we could implement that neuromodulation. Now, you may ask what neuromodulation means in the context of such a simple circuit. Now, one thing that we want that is key to neuromodulation, especially when we talk about these inhibitory circuits, is the fact that by modulating the neuron from burst excitability to spike excitability, so those two modes that I expressed initially, essentially you can decide very dynamically and very easily to disconnect the neuron, a node, from the circuit. And that means that, for instance, in the context of a half center oscillator, you have a control of the rhythm from each neuron. So by modulating each neuron, you can decide whether the neuron will participate in the circuit rhythm or instead will live its independent life. Now, that's extremely convenient control principle when you go from a single cell to a circuit, to have a sort of a dynamic principle that allows you to, for any node of the network, to engage or disengage from the circuit. That's a central feature of neuromodulation in central pattern generators like those that Yves Marder has been studying. And we wanted to replicate this in the half center oscillator. So when you are in the bursting mode at the cellular level, you really engage the classical rhythm between the two neurons. But when you are in the off mode, you can have one neuron spiking and the other neuron being silent. So essentially they are disconnected from each other.
But what controls the connection between them again? Just your
okay,
knob.
So this is the half center oscillator. So you just have an inhibitory connection between the two neurons. Now, usually when people tune that circuits, they tune the synaptic weights. So here instead, we don't touch the synaptic weights. And for a fixed topology, we show that the cellular control can disengage or engage the rhythm, which is a very
How do you control it? I still don't understand. What is the actual coupling control?
What is the control? So how do we control that the cell engage or disengage? Yeah. So this is through neuromodulation. And neuromodulation is tuning the maximal conductance of one specific current source.
Okay.
It is just actuating the base currents of any of a specific transistor. So this is in principle something that we can do at the level of every node of a circuit, okay, if we can control the base currents of the transistor. Okay, so it's a very basic control mechanism of a circuit, but a very important one, we think, and one that has been completely, I don't think there has been much attention to that type of
so Rodolphe, yes? Someone is asking whether, is this an Izzy-Kevich type neuron or not?
It's not. So Izzy-Kevich model is a good example of a model that, okay, you write differential equations of a model, but you don't really have a circuit realization of that model. Or you can work on a circuit realization of that model, but you won't have this conductance-based principle that of having a circuit representation being a single capacitor and then a series of parallel current sources. So this is very biophysical. You don't have this sort of decomposition in a model like Izzy-Kevich. So it doesn't mean that you cannot use Izzy-Kevich model to produce bursting or to produce spiking for specific values of the parameter, but it will be very difficult to relate the tuning of the parameters in izzy model to a biophysical tuning mechanism like neuromodulation.
How about, this is Andreas, how about the Mihala's nibble model?
Likewise. So, so I would say that any model that has been proposed in the literature and that has the structure of the conductance-based model, so that has the circuit realization of a conductance-based model with parallel current sources, potentially you could implement neuromodulation and try to use the maximal conductance as your control parameters. Those are the only control parameters that we tune. We don't tune the time constants, we don't tune the activation, half constant of activation and so on. I mean, this is given, this is frozen for the architecture of a given neuron. So once we have a given neuron, the only thing that we tune are the maximal conductance. And we show that if the neuron contains the right current sources, you can really span the whole set of patterns that you would like to see in a classical neuron just by tuning those maximal conductances. So we try to be consistent with the principle of, with the biophysical principle of neuromodulation. In neuromodulation, there is very little evidence that time constants are changing, or that half constant activation are changing. But there is a lot of evidence that the maximal conductance, that is the channel expression, is constantly changing and adapted. Does that answer the question?
Yeah, so please define what you mean by neuromodulation. You mean by the fact that, that the entire neuron or class of synapses are all increased or all decreased, or something like that. Is that what you mean by a neuromodulation mechanism?
So each current source is essentially a voltage to current relation, very similar to the intrinsic current. Okay. Now, typically you model a Hodgkin-Huxley current with an activation time constant and then with an activation range. Okay. This is the same thing. And then it is multiplied by what is called the maximal conductance. So when all the gates are open, what is the maximal conductance that you can get from these particular channels? This is the control parameter.
And I have a follow-up question, this is Andreas. So all the molecular level mechanisms are basically reflected in the currents, and that's really the premise here.
Exactly, yeah. So it's sort of a way of aggregating a lot of molecular effects, and you can change the expression of channels, you can change the susceptibility of receptors. There are lots of different mechanisms at the molecular level that can lead at the macro level, at the cellular level, as changing the maximal conductance.
And you don't lose anything by ignoring the dynamics of the molecular level?
Well, that will reflect in how fast you change your maximal conductance, right? So you should, so here, I mean,
Thank you.
Yeah. Shall I continue?
Yeah, please. And we're about 10 minutes. We're going to be about 10 minutes over, which is fine, right? But let's just go on and
okay.
And
okay. So I spoke about a very specific control problem, right? Which probably is reminiscent from the very early days of Telluride. I think when Telluride mentioned examples yesterday, I think neuromorphs were interested in CPGs probably in the 90s, but not much later than that.
1980s.
80s. So I'm sorry for talking about history here, but we like to revisit history, and we hope that it leads to something novel. Now, maybe I'd like to scale up and to give you for 5 minutes the big picture of what we are aiming at, and what I would call neuromorphic control. So neuromorphic control, so we like to think of a neuronal system as a control system. And what we try to do is really to identify what are the control principles of neuronal systems that are different from the usual control, the principles that we teach in control systems. And then we we ask the question of, can we imitate those neuronal control principles in order to design new control features? We are completing a monograph on this, and anyone is very welcome to receive a draft very soon. So if you would like to give us feedback, it will be highly appreciated. Now I'm going to just give you sort of a snapshot of the approach in this monograph. This is the block diagram representation of a neuronal model. I don't know if you have ever seen a control system, you might have seen such a block diagram. So we think of the passive membrane as the plants to be controlled. And then we think of the ion channels as being the controller that we design. And then we think the external current as affecting the passive membrane and changing the voltage. And the design of the ion channels will sort of shape how the voltage is sensitive to a change of the external current. Okay. Now, this model is nothing new. It's just a reinterpretation of Kirchhoff's laws of Hodgkin-Huxley model. But it sort of highlights the fact that the only thing that feedback can do in a control system is to shape sensitivity. And by sensitivity, I mean, if I change the input here, will the output be sensitive to that change? Now, if you compare this to the cruise control system I was referring at the beginning of my talk, there is a very strong analogy. That's a very classical control system that we teach in Control 101. So we teach that if you have a car and want to regulate the speed of the car, you design a control that is based on the velocity, a measurement of the velocity will produce some internal force from the engine that will try to make the velocity insensitive to external disturbance, like the load of the road. Okay. Now there is a very strong analogy because, you know, the electrical model of the passive membrane is exactly the same as the car model. So if you use a force velocity analogy, the force velocity model is just a mechanical analog of the current voltage model. And so you can really ask a question, if you ask how different is a neuron from a cruise control system, is how different is the cruise control from the ion channels? And the big difference is in the response, right? If you have a bump in a road, or if you have a bump of applied currents, in open loop, you will just have a passive response. So you will have a bump in the velocity or a bump in the voltage. In closed loop, the cruise control is designed to attenuate the sensitivity, to reduce the sensitivity, because you want to make the velocity insensitive to a bump in the road. Instead, as we know in neurons, if you exceed some threshold, you trigger a spike. And a spike is nothing but the manifestation of an ultrasensitive response to a disturbance. And that's sort of the starting point. And then the second observation is that the only way to achieve an ultrasensitive response in a robust way that is in the presence of uncertainty is to use feedback and to mix feedback. So to use a mixture of positive and negative feedback. And now we can sort of attack the design of a neuron and as thinking of designing a controller that has to balance one source of positive conductance and one source of negative conductance. And that's exactly what ion channels do. And we get ultrasensitivity whenever we have a balance, an exact balance between the positive conductance and the negative conductance to get a point of zero conductance. So there is a mathematical equivalence between a point of zero conductance in this circuit and ultrasensitivity from the current to the voltage. So we reinterpret excitability as a very robust mechanism to achieve ultrasensitivity. And from there, we try to scale up. So we say, if we go from one neuron to a whole field, perhaps what conductance do at every scale is exactly trying to maintain this balance and to create local scales of ultrasensitivity. By this, I mean that the ultrasensitivity is very localized in every possible sense. It happens at a very specific voltage. So it happens in a very specific range, but it also happens at a very specific time scale. So it only happens if you excite the neuron with the right dynamics, with the right scale. And it also in space only happens at a very specific spatial scale. And by combining many local scales, you can build broader scales and you get a multi-scale field. And we think of control as shaping the sensitivity of a spatial temporal electrical field. And all of this is done by adjusting and by tuning this balance of conductance at every possible level. And the specific emphasis of neuromodulation is that we put as much emphasis on the intrinsic conductance, so the conductance that are intrinsic to the neuron, and the synaptic conductance, so the extrinsic conductance between neurons. And that's a striking difference with respect to most of what I've seen in neuromorphic design and neuromorphic computing systems, where there is a lot of emphasis on synaptic tuning, but very little on intrinsic conductance tuning. But in fact, the lesson from neuromodulation is that for a given synaptic topology, you have all possible behaviors just by changing the internal conductances.
Okay.
I think I should wrap up. So this is just to tell you that at the end of the day, when you go from this very simplistic single input, single output design, you can scale it up and now think of the external current as being a spatial temporal field, and this voltage as being a spatial temporal field. And the passive behavior is essentially an RC network. That's a physical substrate. And then the controller is a fading memory that controls all these and balances all these conductances from any neuron to any other neuron. Now this feedback loop is you can think of the fading memory as an artificial neural network. You can think of the fading memory as a deep neural net, typically feed-forward deep neural net. So this architecture is a very specific type of recurrent neural network. But it is very consistent with the biophysics of any neuronal system. And I think that the key difference between what we have learned in tuning deep neural nets and what we understand today in learning is essentially the tuning of the fading memory. But what is ahead of us is to understand how you can leverage the tuning of that fading memory in this particular feedback configuration and use that learning to tune the sensitivity of a field as opposed to do a classification task. And I'll finish with a proposal for perhaps something that could be a Telluride challenge next year. And it would be to scale up. So right now we have understood the design of that fading memory, either for a single neuron or for very small circuits of 2 or 3 neurons. But what if we scale up to 20 and maybe to have hundreds? So right now we are working with Gert Cohenbergs on designing a neuromorphic architecture that will allow a neuromorphic chip of a few hundreds neurons to have these tunability properties. And we want to think of simple tasks that will demonstrate the potential of tuning not just the synaptic conductance, but also tuning the intrinsic conductance. And to do that in a principled way, so instead of starting from, let's say, reservoir computing, like what we will hear in 2 days from now, it's really a bottom-up architecture where we always use conductance by pairs. We always balance a positive conductance with negative conductance. And we have a very specific tree architecture in space, in time, in amplitude to organize the feed-forward network of these mixed conductances. And just to finish, this is an illustration of a 5 neuron network So it's very much like coupling two half-center oscillator with a central node. And this is a cartoon of the network that Yves Marder has been studying for 40 years. And one of these half-center oscillator has a very slow rhythm. The other one has a fast rhythm. And so here you have essentially a network architecture that produces two distinct rhythms and that can combine those rhythms in any possible way. Now, if you try to tune any of those to produce, let's say, the rhythm that you see in purple, if you try to do that only by tuning the synaptic weights, it's a nightmare. It's very fragile, it's very unrobust. And instead, just using this idea that I talked about earlier, that any neuron can engage or disengage the network by being neuromodulated, you go very easily from a network where the 5 neurons interact with each other to a network where the 2 rhythms are completely independent of each other in any intermediate situation. So that's an example of how we could combine, you know, all what you guys are doing with neuromorphic design, but adding this additional feature of having a very dynamic control of the functional connectivity of a neuron without touching the anatomy or the synaptic connectivity of the network.